Hello, welcome to Mr. Sauceda's YouTube videos. Today, we're going to be talking about Lesson 4 of Genetics, which is how do you make and interpret genetic disorder Punnett squares. So we spent a lot of time looking at simple Punnett squares, which, by the way, are exactly the way that you make genetic disorder Punnett squares. But there are two categories of genetic disorder Punnett square that we're going to be talking about, so it's a little different. Okay, first things first, how do genetic disorders work? We need to talk about chromosomes to some degree. So humans typically have 46 chromosomes. That's 23 pairs of chromosomes. We already talked about that when we were doing mitosis a long time ago. The first 22 pairs, though, are called your autosomes. Auto means self and some means body. So they're like the ones that code for like your body. They code for the majority of the things that you see. Here are the first 22 pairs of chromosomes. Kind of organized, by the way. Like this is chromosome 1, then 2, then 3, etc. The last pair, those are called your sex chromosomes. So here's an X and here's a Y. The sex chromosomes, when we talk about non-disjunction disorders in the next lesson, are going to be kind of important. So they're kind of separated, though, from the autosomes when we're talking about genetic disorders because they're kind of separate and special and have their own kind of issues and stuff when we're talking about genetic disorders. All right, so keep in mind, right, in humans, there are two sex chromosomes. We have the X chromosome, we have the Y chromosome. XX would be assigned female at birth typically, and then XY would be assigned male at birth typically. However, that isn't always the case, and that's because like the Y chromosome can have like some chromosomal issues, which I guess I'll show probably a little bit. It's the SRY gene, which means that you could be XY and assigned female at birth. Anyway, we'll talk about the intricacies a little bit, but just kind of know XX is typically assigned female, XY is typically assigned male. Okay. Now, this is our system right here. So XY, that symbol, by the way, is male, and that symbol, by the way, is female, in case you've never really seen those symbols before. But what do you notice about these other kind of organisms and these other kind of sex-determining systems that other animals kind of have here, right? So take a look at it. You can pause it if you wish. Okay, let's talk about these birds. So in these birds, you can see, right, it takes two copies of the same chromosome to be male. So you can kind of think of this as like the inverse of our system, right? Where they call it the ZW. So instead of X and Y, they call them Z chromosomes and Y chromosomes, just because like it's kind of the opposite of our system. So in order to be female, you have to have the two different chromosomes. Whereas on our version of the sex determination system, if we have two different chromosomes, we'd be assigned male, right? So like, and that's typical in birds. So you see it a lot in birds. Here's another interesting one with mice. So some organisms don't just have two sex determining chromosomes, they have 3 or more. So this one's 3. And so again, in order to be male, this mouse has to have an X and a Y. But like if you have a Y chromosome, that doesn't mean that you're male. You have to have both the X and the Y. So you can have a W chromosome and a Y chromosome and still be phenotypically female. And so this one is even more complicated. Like you have 3 different chromosomes that code for sex-determining characteristics. And this one is even crazier. This fish one, you have 4 chromosomes that are getting inherited that determine biological sex. And there are so many differences here. And it's like there's this gene that can get passed around between chromosomes, right? So just keep in mind that our system is relatively simple compared to a lot of other organisms on Earth where they get very, very complicated. Now here is an X chromosome versus a Y chromosome. The Y chromosome is tiny, and this SRY little gene here at the top is typically what, if there are issues or that gene is missing, then you would not be assigned male even though you have a Y chromosome. Okay. Now, in this unit, we are purely talking about biological sex. That is literally it. Biological sex refers to the reproductive organs, the hormones that a person has, right? It's typically thought of as binary. However, it is not binary. There are intersex individuals, right? Intersex individuals are individuals that are born with ambiguous outer genitalia and parts. Or they could have both sperm producing and egg producing cells, which technically is called true hermaphroditism if they can produce both. But, like, again, it's just way more complicated. Now, this is not a sociology class. This is not a psychology class. So we're not going to be talking about like gender, which is socially constructed and culturally constructed. We're only talking about biological sex, okay? Which again is not technically binary, and neither is gender, but gender is more complicated. Okay, so let's talk about autosomal disorders. Autosomal disorders are caused by mutations in the first 22 pairs of chromosomes. We're not talking about sex chromosomes at all. The first category are called autosomal recessive disorders. Now, autosomal recessive disorders, in order to have that genetic disorder, you have to be homozygous recessive. That is the only way for you to have the disorder. So you might think to yourself, well, what if you are heterozygous, right? What if you have one copy? That makes you a carrier of the disease. You do not have the disease, but you are a carrier of the disease. Carriers are super interesting because they have no symptoms. They typically have zero, like unless you looked at their DNA, you'd have no way of knowing that they have this allele. But they can pass on that mutated allele to their children. So we have two carrier parents. That's why they're kind of different colors here, like they're split orange and blue. So we have two parents that are carriers of a genetic disorder. What's the likelihood that their children would be born with the genetic disorder? So 25% chance that they have unaffected children, 50% chance they're carriers, like their children are carriers. Again, they're unaffected though, because carriers are unaffected by the disease. But there's a 25% chance that you have a child with that particular genetic disease. So what's a good example of a genetic disease that is autosomal recessive? Cystic fibrosis is probably the most common one that everybody uses as an example. So what is cystic fibrosis? It affects the lungs. It is typically caused by literally a single problem with a gene on your chromosome number 7. So your 7th chromosome, there's a gene. That gene has an issue. Now, what does that gene code for? It codes for in your cell membrane. So remember, we have phospholipids and you have your protein channels. It codes for an error or a problem with a protein channel. This protein channel is letting chloride ions go in and out, and it's not letting it do it correctly because it's misshapen. And so by doing that, it literally causes issues with your lungs so that you're building up mucus in your lungs now. Okay, so let's do a Punnett square for that. Capital C is normal, lowercase c is cystic fibrosis. Okay, both parents are carriers, so we're gonna have a capital letter C and a lowercase letter c. Capital letter C, lowercase letter c. This child would have capital letters only. These two children would have one capital letter, one lowercase letter. These two children would have two lowercase letters. So when we're doing our breakdown for our genotype, what percent of children are going to be homozygous dominant? That one child, so 25%. What are going to be heterozygous? These two children, 50%. And then what about homozygous recessive? Just this one child. So 25% chance that your kid has cystic fibrosis, even though you have no symptoms of cystic fibrosis. You might not even know that you have cystic fibrosis allele at all. And there's a 75% chance that your child does not have cystic fibrosis. So if we're coloring them in, there's these three would be not having cystic fibrosis, and this child would have cystic fibrosis. All right. What about an autosomal dominant disorder, right? Like, what about that? So if you have an autosomal dominant disorder, you could be homozygous dominant and have it, or you can be heterozygous and have it. There's no such thing as being a carrier. And so because there's no such thing as being a carrier, these diseases are a little bit rarer, but they're also a lot more dangerous. So here we have an affected father, and technically he's heterozygous, right? But remember, if this were an autosomal recessive disorder, he would be a carrier. But because there's no such thing as a carrier, he's going to be affected by this disease. The mother, on the other hand, two lowercase letters, so she's not going to have the disease. What's the likelihood that you're going to have a child, though, with the disease if one parent has it and the other parent doesn't, like this? There's a 50% chance. And if this parent was homozygous dominant, there would be a 100% chance that the children would have it. So again, that's why they're much less common and also a little bit more dangerous. So what's a good example of a homozygous dominant disorder? Huntington's disease. Now, Huntington's disease is really dangerous because it's often a disease that you don't know you have. So you could be born with it, not know you have it until you're in your 40s or up to your 60s. And the symptoms are very, like they seem like Alzheimer's. They seem like, in some cases, natural signs of aging where you're forgetting things. And what makes it dangerous is that if you're in your 40s or your 60s when you have this, you probably have already had like children and may have already passed on that particular gene or allele to them. So let's pretend that we're drawing a Punnett square for an autosomal dominant disorder. So capital H is Huntington's disease. Remember, it's dominant, so the capital letter is the one with the disease for this one. Lowercase letter h is going to be normal. Only one parent is affected. Okay. So we have, again, a capital letter H and a lowercase letter h. The other parent, unaffected, two lowercase letter h's. These two children are going to have Huntington's disease. These two children are not going to have Huntington's disease. So there's a 0% chance of having a homozygous dominant child, but there's a 50% chance of having a heterogeneous child. There's a 50% chance of having a homozygous recessive child. That means that there is a 50% chance these two children are going to have Huntington's disease. There's a 50% chance these two children are not going to have Huntington's disease. All right, so that's it. If you have any questions, let me know. Make sure you fill in the notes as you were going through there.